play with Rousseau? How does our get started this one? All the concerto for two trumpets. Like cool little thing, and I was a sophomore. And I was, in all of the masters there. And he was going, "What are you playing on there?" And I had my and he left, and then my band, college band director came over. He goes, Dean wants you to make, designer of them. He's like, he wants you to, so I've been on them ever since.
I study with Bishop. We study with him. Oh, so yeah, so it worked out since I had him, but I tried a few custom EXs when I was getting my tenor. Like, each one played different. Got a really good deal with this and my yeah. Got kind of both of them. So yeah, I got it. It's always nice to have your own horns.
Last year, you saw the Rosewood Brothers. They were playing. They were 14, 15, two guys. They play going. They wear hats and they would act. Yeah. And so Marshall has them come out to the Creative Arts Foundation. They have brand new horns. My husband died. And a brand new Yamaha. I got one of them. I was like, you know they want me to buy with old school? They don't need a lot. I can just go with it. Like, nope, we're just dealing over 14-year-old.
Half of it's the act, play and then
Joe, you guys ready? No, you're good.
just hype up the audience and make it hotter.
Ladies and gentlemen, put your hands together for the Space Coast Little Big Band. All right, everybody. What a bunch of talent. Thank you all for coming to the first possible annual Animal Talks. I don't know, we were talking about it might be Animals Keep Talking next year. I don't know. I'm your host, Jared Campbell, and I would like to thank you all for coming. But enough about me. We're here to really celebrate Phi Kappa Phi in a way, and also just intelligent things in general. But first, before we get into any of all that stuff, I would like to introduce you to Dr. Heidi Edwards, the president of the Florida chapter of FIT's Phi Kappa Phi.
Thank you, Jared. Welcome to the first of what we do hope, as Jared says, is an annual event celebrating research and Florida Tech scholars who live the Phi Kappa Phi motto, let the love of learning rule humanity. I'm Heidi Hatfield Edwards, as he said, president of Florida Tech's Phi Kappa Phi chapter. The Multidisciplinary National Honor Society recognizes and promotes academic excellence in all fields of higher education and engages the community of scholars in service to others. Your Phi Kappa Phi chapter is active and engaged. This year, we were recognized as a Circle of Excellence Gold chapter and recently won an award for our fall service project, making oyster mats for the Indian River Lagoon Research Institute Living Docs project. PKP's focus is on literacy, and our project promoted environmental literacy through an on-campus event and social media promotion. Our success is due to the dedication of our members and our executive board. In particular, the Fall Service Project would not have been so successful without the leadership of our student vice presidents, Angelica Zamora-Duran, Kaglar Erdogan, Ryan Fink, Donovan Southwell. Each year, we invite the top Florida Tech scholars at the undergraduate and graduate levels to join Phi Kappa Phi. Those invitations will go out soon, so be on the lookout. Membership may be its own reward, but the opportunities for scholarships and fellowships and other benefits are pretty good too. Our chapter has had really good success. For example, last fall, one of our members, Kaylee Koval, received a $500 Love of Learning Award from the National Honor Society. We are blessed to be on a campus full of people, all of you, who truly love to learn, love to serve the community, and importantly for tonight's event, love to have fun. So let's have fun, enjoy the evening, and let the love of learning rule humanity.
Pretty good. These guys are amazing, super amazing. You know what I've always wanted to do? I've always wanted to do something like James Brown. If I say, hit me once, I say hit me twice. Bring it home, baby. Thanks. Well, so here we are. I would like to talk to you a little bit about wait a minute, that's not an animal. That's not animals at all. Oh, my goodness. Oh. So, Dr. Drew Palmer is going to be speaking here in a minute. I'm supposed to stand on the X. Science at the interface of chemistry and biology is the driving theme of Dr. Palmer's research. Whether it is developing a plan for growing food on Mars, deciphering the chemical signals that bacteria use to cause disease, or understanding what makes algae swim. His research is at the intersection of the natural sciences. He grew up in St. Augustine. He received an AS from Tallahassee Community College, a BA from Florida State University in biochemistry, and a PhD in biomolecular chemistry from Emory University. And he then did a National Institute of Health postdoctoral fellowship at the University of Wisconsin-Madison. His hobbies include cooking, home brewing, and making coffee. Ladies and gentlemen, give it up for our first speaker, Dr. Drew Palmer.
Hey, I have that. Clicker. Thanks. So I want to thank Jared in particular for giving me an opportunity to use my dad joke for the night. So I'm actually a plant here tonight. Okay. So now that I got that out of the way, I can start giving a real talk. So what I'd like to talk to you guys today is about the secret social life of plants. And when I was thinking about this and what makes plants interesting and how I could kind of fit into all of this, is you have to start by thinking about what do we mean when we say a society. So what's a society? So I kind of found this image on the web and kind of did a little Google definition search, and it's an aggregate of people living together in a more or less ordered community. In my house, it is probably a less ordered community, a far less ordered community. And there's a lot of benefits to being in a society that we can agree on. And one of the major things about being in a society, though, is that in order to function, you usually have to have rules, and you have to have some kind of communication between the participants in the society, right? And so are human beings the only ones that make societies? So typically we'd say, yeah, if you don't think about it much, you'd say, yeah, human beings, we make societies, right? That's what makes humans unique. But what about all these things, right? A pride of lions, a pod of dolphins, one of my favorites, the murder of crows, apparently what's called a flamboyance of flamingos, right? And so all of these groups have social interactions of their own. They have their own ways of communicating. They have their own social structures and hierarchies. Even down, if we look at these, I'm used to having a laser pointer, so excuse me. If we look at these termite mounds here, right, that you can find throughout the world, these termite mounds are built by industrious termites that actually have division of labor. There are ants that are responsible, or termites that are responsible for building the mound, ones that go and collect and forage. There are defenders of the mound. So there's a highly organized division of labor. In fact, those kind of observations led this guy, a biologist by the name of E.O. Wilson, to kind of envision a new field of biology called sociobiology. And there's this nice fancy quote here. But really what sociobiology is looking at the animal world and seeing the different kinds of social behaviors that have evolved out there. And saying, okay, what we do as human beings is one way of running a society, one way of organizing social structures. But evolution's found lots of different ways to do that over time, right? And so this last part's what I like. By looking over the realized social systems of hundreds of animal species and deriving the principles by which these systems have evolved, we can be certain that all human choices represent only a tiny subset of what's theoretically possible. So what he's saying is, look out in that animal world, see all those possible different ways of organizing things, and then that helps us learn something about our own behavior, right? But also helps us think about other ways of structuring the future, how we want to build societies in the future by learning something from how animals have been successful. Well, obviously, I'm not an animal biologist. I study plants loosely. I'm mostly interested in plants because they do cool chemistry, right? But so how do plants fit into this, right? Because obviously, right, these things are social, right? The things on the right side here are social animals, right? And this thing on the left, this big redwood tree, they're not social, right? Plants can't be social. They sit around all day. They don't talk to each other. What would they talk about, right? There's a lot of rain today, right? So they're just sitting there being tall with all their photosynthesizing, right? But And we think they don't have any of these senses, right? They don't have sight, they don't have sound, they don't touch each other, they don't taste, they don't smell, or do they, right? So I'm just going to start with one example. So what about the sense of touch? This is a tiny little plant, the mimosa. And these mimosas, if you touch the mimosa enough times, you touch the leaf in the right way, that leaf will actually curl up, right? Same is true of Venus flytrap, right? That's responding to touch, right? So to some limited degree, plants have the ability to respond to touch. Now, if you want to see a really interesting experiment, when I first started teaching at Florida Tech, I'm going to give you a little anecdote here. I sent out students to do a scavenger hunt in a plant science class. And out by the Scott Center, there's a mimosa, except it's called a false mimosa, which means it doesn't do this. So I told them to take a picture of them making it do that. And they were like, Dr. Palmer, I spent 2 hours stroking this flower and it never happened. So I'm going to blame the plant for that. It knew better. All right. So what about taste and smell? Can plants taste? Can plants smell? All right. Well, I'm done. So what is the sense of smell? What is your sense of taste, right? This is really, and this is where I come back to my chemistry background, is that all these are is the ability to detect molecules that are floating in the air or in water, that's, in the food that you eat, right? So for instance, this molecule on the top is called methyl salicylate. It's what we associate with the smell of wintergreen, right? While the one on the bottom is vanillin. I'm standing in the way of them right there, right? And so vanillin is the smell that you associate with vanilla, right? And all that happens is if you look in your head, right, you have these little cells that work as receptors for these different molecules. These molecules bind to the receptor like a microphone picking up a signal, and it transmits it to your brain and translates that, right? So these guys have cells for this. We have cells for this. Can't plants have sense of smell? Can't plants have a sense of taste? So sure, right? So one of the best examples is what we'll call the synchronization of fruit ripening, right? Or, hey, put those fruits in a brown bag together, right? You've heard that before. You want to get something to ripen, you put it in there with another fruit. The reason why is plants release something called ethylene. It's a gas, and that gas actually diffuses out, and it allows fruits to essentially synchronize when they're going to ripen. And that means that the plants in the area can maximize when animals are going to come and help distribute the next generation of fruits, right? That's pretty good. So then this is the black walnut tree, and it will release into its roots this molecule over here, which is called juglone. And what it actually does is it says, hey, this is my space. And it tells other plants, don't grow here. So other trees won't start growing and trying to fight for space. It kind of marks its territory, right? And that's just something that's in the ground that other plants can detect. So this is one that's one of my personal favorites. This is by a gentleman by the name of Ian Baldwin, who's at the Max Planck Institute. And Ian's work is looking at how plants respond to when herbivores come and kind of munch on them. So this is sagebrush. And Ian noticed that he could put sagebrush kind of physically near each other, and he'd fence one off where so animals couldn't get to them, and he let animals feed on the ones over here. And he trapped molecules being released by the plants getting eaten that basically communicate to the other plants and say, don't grow as tall, right? Maybe if you stay small, they won't notice you, right? And so this is over a fairly small distance, but it still allows them that one opportunity, right? This is an even cooler one. This is Junji Takabashi. This work is looking at tomato plants and caterpillars. And when caterpillars eat on the tomato leaves, the tomatoes will release a signal that travels through the air. It's what we call a volatile signal. So it travels through the air, and then the tomato plant next to it picks up on that signal and says, there's a caterpillar around. I'm going to change the chemical composition of my leaf just a little bit so I'm a little less tasty. And in fact, if any of you home gardeners have ever noticed your insects switching hosts during the year, that's actually a well-established phenomenon, that plants will allow themselves to be eaten for a certain period of time, and then they say, yeah, I'm done with this. And they release chemicals that push things like aphids to a different plant. All right, so I'm a scientist. I can't get away with not talking a little bit about my own work. I became really interested in this phenomenon because of something called kin recognition. It was that someone told me that plants that are closely related to each other can actually recognize like their siblings. And I went, no, that's not a thing. And so we started doing some work in this area. And so the whole point is that plants do this to basically figure out how they're going to compete for nutrients in the soil, right? So they're having an active conversation about it, right? And so we're going to set up a couple of systems here. We have what's called the solo plant. So this is the guy living by himself, right? The eternal bachelor, right? And then we're going to put together what we call kin. And the kin plants are plants that are of the same, what we like to have a fancy word for called ecotype. You can think of it as breed of dog, right? These are just two different breeds of the same plant, right? So, or these are both of the same breed. This is what we call Columbia Zero, right? And then over here we got what's called stranger. And stranger means that these are two different breeds, right? They've never encountered each other before, right? We did a little test. We're going to grow them in a pot with each other. We're going to say, okay, how do you guys interact with one another, right? And so little data to round out the night. What we found is that if you have the plant by itself, right, what we're counting are the number of roots that it forms, right, how many roots it branches. Because the more roots that it makes, the more likely it's competing for nutrients, right? What we found is that if you have a really lot of nutrients, right, so over here on this side we got the 1. That means the most amount of nutrients. And then we're going to go backwards to the lowest amount of nutrients, right? So the solo, which is the plant by itself, our bachelor, right? He's got plenty of nutrients. He doesn't make a lot of roots. The two plants that know each other, they're same breed, they're a little bit more. Stranger plants both have a lot more lateral roots, more roots, right? So they're starting to compete. And as we actually reduce the amount of nutrients that are around, so we're going this way, right? We're going back. You can see that they all start to make more roots. Remember, the number of roots is something about the amount of competition that's going on, right? So it turns out that these plants can actually tell the difference between plants that are of the same breed, right, and different ones. They're able to recognize that much and decide that if it's the same breed, they want to compete a little bit less, right? It's like recognizing a member of the pack, right? And so you can actually trick the plant. And so we can take the dirt we grow the plants in, we can do some fancy chemical extractions, just like brewing a coffee, right, from grounds. And then we can put those back. And if we put those on the plants, so in this graph what we're doing here is we're taking a solo plant by itself, and we take its dirt and we put it back on it, the stuff we isolate, and it doesn't change much. We put that kin, so its friend, and it grows a little bit more. We do the stranger and it grows even more. So there's a chemical signal in that dirt that we can extract and then we can make work for us, right? And so this is what we're trying to do, is we're trying to learn how to translate the languages that all these plants talk. And so then we can use it, right? We can learn how do we compete, how do we help regulate how much nutrients we need? How much fertilizer do we need to put in our yard, right, in order to get the plants to grow better? Right? So, okay, does this have anything to do, plants can talk to each other, but surely they can't talk to the rest of the world, right? Well, actually, plants are really good at talking to the rest of the world. So this thing called tritrophic interactions up here at the top, that you see that little black caterpillar with all these little white stuff sticking out of it? Those little white things are parasitic wasps that have laid eggs on this caterpillar. So this caterpillar made the mistake of eating a plant. And that plant said, I see what you're doing here, and I'm going to release a chemical into the air to attract the wasps that is going to lay all of its eggs on you. And this happens all the time in nature. There's tons of examples of this, right? So plants can even make bacteria work for them. So one of the things we study in my lab is nodulation. So these little bubbles you see here are little houses that the plants make in order to recruit bacteria. And those bacteria are able to take nitrogen out of the atmosphere and fix it like fertilizer for plants to grow on, right? And we've done a little bit of work studying how that timing of that is regulated by the bacteria and the plants and how they work together, right? Over here, mycorrhizal fungi, what you see, this is just the root of the plant right here. All this other stuff is a fungus that grows off out into the soil. And the plant basically takes control of that fungus and makes it grow out and collect nutrients. And that's all regulated by chemical signals, right? And then at the end, this is more of an artistic vision of this, but so if you look at a flower, the thing you're seeing here on the left side, that's what we see, right? And so we go, oh, it's a boring yellow flower. A bee sees the thing on the right. It basically sees a bullseye of where it's supposed to land, right? And so these plants are not just communicating with each other, right? They're not just talking to other plants, right? They're talking to the rest of the living world. They're talking to they're talking to caterpillars, they're talking to wasps, they're talking to pollinators and fungi and bacteria. So they're incredibly social, right? They have all these rules for all these relationships that they're engaged in, and they have all these different ways of communicating with them. But most of them we can't see, right? They're not visible to our naked eye. We can't hear them. We can't smell the signals, right? Most of these chemicals, we can't smell them, right? And so they're invisible to us, right? And so there they are, out there, probably big party animals, right? Hanging out, talking to all these different things, and we're completely unaware of most of it. So, I'll just end by saying you're going to see a lot of really cool animal talks and some very good friends of mine. But, don't forget about your green cousins, So when you're thinking about all these guys, right, think about some of these guys too, all right? So, all right. So thank you very much for your time. And I'm glad to pass this back to Jared.
I need to learn that wasp trick. Ladies and gentlemen, give them up once again for the Space Coast Little Big Band.
No, but I prefer to clip here in my leg.
Ladies and gentlemen. All right, our next speaker is going to talk about something that's, I don't know. It's going to be pretty interesting. I've been waiting for this talk all week. Dr. Nunez is a vascular biologist who uses animal models to study diseases such as hypertension, diabetes, and erectile dysfunction, or ED. One of her favorite projects is the use of a spider toxin to treat ED. This research has been performed in collaboration with scientists in Brazil and is now in clinical trial. In 2009, she started to study how immune system contributes to the development of cardiovascular diseases, which is now being investigated by many scientists all over the world. Currently, her lab is focused on vascular problems and new treatments. Ladies and gentlemen, give it up for Dr. Kenya Nunez.
Oh, thank you. Well, thank you so much for the organizer of this meeting to have me here tonight. It's a pleasure. And I'm very glad to be here. And tonight, I'm going to share with you one story that it's very special for me because it starts during my PhD and is still something that is part of my research, current research in my lab. So today I'm going to talk about spider toxin, erection, and medicine. Well, before I start to talk about my story, I would like to introduce you the main star of my story is the Phoneutron nigriventer spider, also known as a Brazilian wonder spider or banana spider. This spider is not from Brazil, but we have a lot of this spider in Brazil. Brazil is a country in South America, where I come from also. This spider is a very aggressive animal, and rather than building a net, this spider rounds around constantly looking for food and likes to be inside the houses, especially in dark places, to rest. So it's a very dangerous spider. Well, the venom of this spider is, something that I forgot to tell, it's that Phoneutria literally translates for murdery in ancient Greek. So it's a very poisonous spider. Well, the venom of this spider, it's composed by a bunch of substances, small substances that we call toxins. And the venom of this spider, it's 18 more potent than the venom of the black widow spider when tested in animals like rats and mice. And these substances that are in the venom that work as a cocktail, as a mixture of many substances together, can cause a lot of side effects in humans when they are bitten by this spider, including alterations in heartbeat, high blood pressure, low temperature, a condition that we call hyperthermia, blurred vision, and seizures and convulsion. Well, most people don't die because the amount of the toxins in the venom is too small. However, the venom is able to cause another symptom that I'm going to share with you. That's a horrifying symptom. The venom is able to cause intense and painful erection that can last more than 5 hours. The good thing is that it's an erection. The bad thing is that it's very painful. Huh? So, wait a minute. I started to think that if the venom is able to cause intense erection, maybe is there a substance in the venom that could be causing an erection? Then my PhD start. That's me doing my PhD, and very elegant. And the main question at that time was what substance, that toxin, would be responsible for causing an erection. And my goal at that point, I was thinking about that maybe this substance could help men with erectile dysfunction. Well, everybody knows Viagra. Eh? Viagra is awesome. It's in the market, it's FDA approved, has minor side effects, it's all around the world. So why looking for something in the venom to trigger erections? Well, because it's not everybody who answers the therapy with Viagra. We know that many patients, especially hypertensive and diabetic men, type 2 diabetic patients, they have a severe erectile dysfunction, and sometimes they cannot be treated by Viagra or due to the interaction between the drugs, or they do not answer very well the therapy with the Viagra. So before I tell you a little bit about my PhD, I'm a physiologist. I'm going to introduce you very fast to the mechanism of erection. So to make this very simple, okay, I'm going to summarize for you. We have 2 states in the penis that we call flaccid and erect, that to make it very easy, I'm going to call this down and up. So what do you need to know about that? It's that in the down stage, we have a lot of muscle contraction inside the penis and a small blood flow, what is in the majority of the time. In order to have the up stage, the good stage, we must have a muscle relaxation and a lot of blood flow inside the penis. So the blood gets trapped and we have up stage. And another thing is that we need a substance that's very important. It's nitric oxide. So in summary, if we have a lot of nitric oxide, we have a good erection. Well, back to my PhD. So I was looking for, in the venom of the spider, the part of the venom that could cause an erection. After I studied a lot of the time, I found out this part that we call toxin, PNTX2-6, and we tested this toxin in hypertensive and diabetic rats because they are a very good model for erectile dysfunction and mimic very well patients with erectile dysfunction. Then we found that when the rats are treated with this toxin, we got like very good erections and tons of nitric oxide. Okay? One thing that I also found that I'm going to share with you is that my toxin work a little bit different than Viagra. Well, Viagra, it's going to stop one enzyme that's going to release nitric oxide, what is needed for erection. My toxin, it's not going to work via this enzyme, but it is going to induce the release of nitric oxide. So it's as good as Viagra because it's going to release nitric oxide. Well, that is me on the graduation day, jumping. So years later, I found the part of the venom that was responsible for erection. So what we do in academia, we work hard, we found the things, we publish in journals, I graduate, I was very happy. My research became news in ABC News, CNN. Well, spider venom, the next way to treat the impotence. Spider venom, maybe the new Viagra. So we apply a patent for that, trying to use this toxin to make a medicine to treat people with erectile dysfunction. And after I graduated, I moved to the United States, to Medical College of Georgia, and this research didn't stop. We keep doing this research in collaboration with scientists in Brazil, United States, and France. And we still have a problem to solve. We have an erection, however, we have all side effects as well. We have pain. So how we could solve this problem? Well, let me tell you something. Toxin is a kind of a puzzle. It's made by a bunch of small pieces that we can broken down. So what we did, well, a team of scientists, included me, and a bunch of graduate students, we broken down these toxins in small pieces as a puzzle, and each of us started to study this piece, trying to look for what part of the toxin was responsible just for erection, with no side effects. Okay? Then, after a lot of hard work and team work, we found the piece of the toxin that was responsible just for erection. And we call this piece PNPP19. And this, it's me holding the piece 10 years later after graduation. I'm very thankful that I'm still part of this research, and I was able to keep collaborating with the people in this research, and then we found the part that was just responsible for erection. So what we did, everything again, as we do in science. We keep doing things every and every. I grabbed this peptide, the PNPP19, and I tested in rats, also with diabetic, hypertension, and aged rats. As we get older, things go a little bit down. It's not for the majority, but usually it's the life work. But anyway, we got very nice results. The toxin, when injected in these animals, was able to reverse the erectile dysfunction. We observed a lot of nitric oxide. Nitric oxide was released a lot when we are testing this toxin in these animals. And the most important one, no pain, no side effect, just happiness, what we are looking for. So everybody was very happy. So after this work, what we did again? Publish. We published. I was very happy. Our research was highlighted for one of the very prestigious journal in the scientific community, the Nature Reviews. They published about our research Brazilian spider toxin, an analog potentiates erection via nitric oxide. And I was very happy because they make a cartoon with our spider with the colors of the Brazilian flag. And I was very honored with that. And we apply a second patent. Now, to use this small piece that we found in the toxin that was responsible just for erection, but with no side effects, no pain, no major symptoms. Well, just summarizing for you, the story began with me look at the venom and looking for which part of the venom could be causing an erection. During my PhD, I found this part. I identified this toxin that's called PNTX26, and for more than 1 decade, scientists in collaboration, and I'm glad that I'm included, we found the peptide in this toxin that was responsible just for cause erection without side effects. 5 years ago, we started to work with a pharmaceutical industry in Brazil that's called Biozeus. They negotiate the patent with my university in Brazil, and they started to try to put this peptide in a kind of lotion for topical use. And they renamed this PNPP19 as a BZ371. So now, for the venom, from the toxin, from the peptide, we have the BZ, that it's now in lotion and have been tested in men, in normal and in men with erectile dysfunction. And we have got like a very nice results. I'm very happy that we passed clinical trial phase 1. And now we are in clinical trial phase 2. We hope that we survive. And the idea is to have something that can help people with erectile dysfunction and cannot benefit from treatments on the market. Well, that's the story, how my toxin becoming a promising drug, and I hope this to be in the market someday, and I hope be rich as well, as all scientists. Not sure if this is gonna happen. Well, I would like to introduce it, thank you, the people that worked here in my lab, my graduate students in my lab here at FIT, the Laboratory of Vascular Physiology. On the bottom, it's people from my former lab in Brazil, the Laboratory of Venom and Animal Toxins, that have been working on that almost 2 decades. Well, Florida Tech, and thank you so much for listening. It's such a pleasure to talk for our audience, so nice, and I hope that you have enjoyed my story. Thank you.
Ladies and gentlemen, give it up once again for the great Space Ghost with a Big Man. Boy, that last talk has me thinking about Spider-Man for some reason. There's a lot of curious, and you know what? I bet none of those rats could get through the hamster trails. It was funny. It was super funny when I thought it. Our next presenter, Dr. Michael Grace, has been a student of animal behavior since he was old enough to go outdoors. Combining a lifelong passion for outdoor adventure, a deep appreciation of the visual arts, and a commitment to scientific discovery, he built a research program aimed at understanding how animals function in a complex web of interaction among themselves, their potential predators and prey, and potential mates. From snakes to whales, Dr. Grace's group has made numerous exciting discoveries about the nature of vision and non-visual light detection, discoveries that change the way we see the world. Welcome back to the Florida Institute of Technology. Ladies and gentlemen, it's Dr. Michael Grace.
All right, well, thank you, everyone. Good to see you here. So I want to tell you a few stories from the fascinating world of animal behavior. Is that feedback? I hope you find it as fascinating as I do. I've had the good fortune to work with some amazing people and with some truly amazing animals. And so what I thought I'd do here tonight is give you an overview of some of the things that we've been able to accomplish over the years and hopefully excite some of the passions that you have about animals. So, I'm going to talk about animal behavior. Well, what do we normally think of as animal behavior? We think of action. We think of animals actually doing things, like a squirrel gathering nest material to build a nest high up in a tree, or a squirrel running from a person, or not. So, animal behavior is more complicated, though, than just the actions that animals do. It actually starts with animals taking in information from the environment, processing that information, and using it to drive useful behavior. So, for most of my academic career, I've been focused on the ways in which light and things related to light influence animals, their biology, their behavior. So vision is critically important to us and to most other animals on the planet. As an example, imagine what life would be like if we lost our color vision. Or worse, lost our vision altogether. So vision matters. Vision is critically important to us and many other animals. So I want to start by telling you a couple of stories from the world of animal vision. And I want to give you a little bit of background, first of all. In the back of your eye is the retina. The retina is a tiny little wisp of tissue. It's incredibly thin, fairly clear, but it contains millions of cells, including a tremendous number of light-detecting cells known as photoreceptors. And we have two different kinds of photoreceptors in our eyes. We have ones called rods, which are responsible for dim light vision. For example, if you go out for a walk on a moonlit night. And we have cones. Cones operate in high light, daylight, and they give us color vision. So how do they do that? How do they give us color vision? We actually have only one type of rod, so they simply tell you if it's light or not light. But cones, those highlight daytime cells, we have in our eyes 3 different types normally sighted people do. There is one that is maximally sensitive in the blue part of the visible spectrum, one that's maximally sensitive in the green, and one that's maximally sensitive in the red part of the spectrum. And your brain computes how much information is coming in from each of those channels all the time in real time, and that gives us this vast array of colors that we're able to see. So what I want to start off with is to tell you about 2 extremes of vision in the animal world, and actually both come from the ocean. So if you fish, you might recognize this animal. This is the tarpon. It's a mega predator, a massive visually guided predator. And if you look at an adult tarpon's eye up close, you will be amazed. They're about the size of a baseball. They're gigantic. They're much bigger than a cow's eye. Truly a visually guided animal. We began looking into tarpon to try to understand what they see, and how vision develops over the course of time. And it turns out that these tarpon aren't born hundreds of pounds. They actually begin life as extremely small larvae with practically microscopic eyes. And those eyes grow and grow and grow to this massive adult eye. And as they do, they add cells, many cells, millions of cells over time. But what's really fascinating is we found that they begin life only with rod cells, dim light cells. And over the course of life, they add cones. They first add one type, and then another type, and another type. And ultimately, by the time this tarpon is mature, they have at least 5 different kinds of cone cells. So what does that mean? Why should we care? We have 3. Colorblind people, most colorblind people, have only 2. One of them is mutated. In fact, almost all mammals have 2. You can still discriminate color, but the range of color discrimination is rather limited. When you add that 3rd channel in people and great apes, you add a tremendous amount more color discrimination. 4 different types is almost unheard of in the natural world. When we discovered at least 5 in tarpon, we were absolutely amazed. And we estimate that a tarpon, especially down in the blue part of the range, they live in a blue world, have the ability to discriminate color about 10,000 times better than we do. Absolutely amazing. So I told you we were going to talk about extremes of vision. The next animal I want to tell you about is a whale. This is the northern right whale, and that's its beautiful eye there in the center of the photo. It's about the same size as a tarpon eye in an adult northern right whale. The northern right whale is critically endangered. Only about 400 still exist, and their numbers tend to keep declining. It's a sad situation. So we decided to look at the visual system of whales to lend our expertise to try to aid in conservation efforts for these amazing animals. And we were really shocked by what we found. First of all, whales, they have 2 different kinds of cones, but in all whales, one of those cones is inactivated. So whales really only have one functional kind of cone, and they have rods as well. But the amazing thing that we discovered is in the northern right whale and in a few of its relatives, the one remaining type of cone cell that they have is broken. There's a genetic mutation that makes it completely non-functional. So now the northern right whale, this critically endangered species, has only rod cells, those dim light cells, no functional cones whatsoever. Really an amazing story, true extremes of vision, from extraordinary color discrimination to, in these whales, without question, colorblind. And we still don't really understand what they're able to see and how they're able to see. We've done some analysis of the retina, and we believe that those remaining rod cells have taken over some of the cone function. But again, big surprise and true extremes of vision in the animal world. And we, people, lie somewhere right in the middle between these two. All right, so now for something completely different. Snakes. People always want to say, why did it have to be snakes? So I could have spent the entire time talking about snakes. I love snakes. They're fascinating animals. Their biology is amazing in so many different ways. Snakes have eyes. They can see. You can see the eye in this animal. But some snakes, not all snakes, but a very few snakes, including pythons, boas, and pit vipers, have an extraordinarily amazing, unique, vision-like sense. They have what are called pit organs in their faces. They work similar to the way eyes work. They gather photons, they gather electromagnetic radiation from the environment, and they convert it into an image in the brain, much like the visual system works. But what's different about those pit organs, their eyes look at the visible world just like we do. The pit organs detect infrared radiation, far beyond the red end of the visible light spectrum. There is no other animal on the face of this planet that does anything like this. Truly, amazingly unique. In fact, we have developed a lot of artificial infrared detectors for a lot of different purposes, in your home, in industry, in medicine, lots and lots of different purposes. Nothing, natural or artificial, comes anywhere near close to the value and the functionality of the infrared imaging system in these snakes. So, in the interest of time, I'm going to leave those snakes because I want to tell you about another completely different use that animals have for light. So some animals can change color and pattern in dramatic ways. Think of chameleons, octopus, cuttlefish. I don't know if you've seen these videos. Some of these creatures, they will just shimmer with color and pattern as they just change. They can match their backgrounds in amazingly precise ways. No one has any idea how this works until now. So I hope everybody signed the waiver on the way in because this is brand new. We've never talked about this before in a public forum. But we studied one of these color-changing animals that also lives right out here off of our coast. It's called the hogfish, another one people may fish for. The hogfish has the ability to change color and pattern in pretty dramatic ways. And we wanted to try to determine how they do it. Now, there's a problem for a hogfish. What's he trying to do? The fish is trying to change color, change pattern, perhaps to impress a girlfriend or a boyfriend, or to camouflage itself against potential predators, or maybe to sneak up on prey. But how do they know what they look like? A hogfish, trust me, it cannot turn its head and look at its body. It can't do it. They can't hold up an arm and say, what am I looking like? And as far as I know, there are no mirrors out there in the ocean. So how do they know? Well, if you look at the skin of a hogfish, the first thing that strikes you is this array of color-changing pigment cells. They're called chromatophores. And they are responsible for the color and pattern changes in these animals. But what we recently discovered is that underneath those color-changing cells, directly underneath those cells, is an array all over the body of thousands of light sensors, photoreceptors, spread out all over the skin of this animal. And so what we think is happening in these animals is they use their eyes to look at the world and understand what's happening around them, the background, is it a coral reef, is there a predator, is there prey, is there a potential mate? And then they use these light detectors in the skin to sort of look out through what I like to think of as sort of sunglasses, color-changing sunglasses that are these pigment cells. So in real time, I'm swimming around the reef, I see the world around me, I know what I want to be, and I ask myself what the world, what I look like with these light sensors in my skin that are looking through those pigment cells. Really an amazing, amazing system. All right, so I've told you about tarpon, amazing color discrimination. Whales, zero color discrimination. I've told you about snakes, looking at the world in a completely different part of the electromagnetic spectrum. And hogfish, ability to look out through their skin and understand themselves in real time. So the last thing I want to do, hopefully I can do this efficiently, it's a little challenging, but I want to tell you about a bigger, broader effect that light has on animals. We have rhythms of lots of things in us all the time, every day, circadian rhythms. When we sleep, when we wake, when we're hungry, our blood pressure goes up and down on a daily basis. Our body temperature goes up and down. Hormones fluctuate on a daily basis. These are all driven by internal clocks, circadian clocks, molecular clocks in our brains, in other animals, in other parts of the body. And those clocks are profoundly affected by light, naturally by the daily light-dark cycle. That's a good thing. The problem, and people are starting to recognize this as a real problem, is artificial light at night. We began, as part of the study on tarpon, we started looking at tarpon from some of the coastal areas around here and south of here, areas where we call them nursery habitats, where these tiny little fish develop and ultimately move out into the ocean, repopulate the ocean year after year. So these nursery habitats are critically important, but they are precisely the places that people also love. Places like Biscayne Bay, Tampa Bay, even the Indian River Lagoon, and many more places like this around the world are subject to tremendous levels of light pollution today. We discovered recently that these circadian rhythms that we see in land animals, they also exist in these marine animals, marine fish certainly, and almost certainly lots of others. There have been some interesting studies that show that people and animals that are exposed chronically to artificial light at night suffer significantly higher health problems and have shorter lifespans, immune system compromise, on and on and on, lots and lots of troubles. So we're very interested in how this artificial light at night affects these animals in the natural world. So let me end tonight by sharing a quote by one of my heroes. George Wald won the Nobel Prize over 50 years ago for discovering the molecules that are the first step in seeing, the molecules. And those molecules have driven essentially everything that I've told you about tonight in my career. So he wrote in a talk recently, near the end of his life, the retina of a frog is very much like a human retina. Both contain light receptors. The nervous connections to the brain are much alike. But I know what I see. Does a frog know what he sees? I say yes. Frogs do know what they see. They understand it. They make decisions based upon it. I think George Wald would be amazed if he knew all of the developments in the interactions between light and animals that have come about since that time. There are tremendous practical applications, but I think the primary reason for loving this and for doing this kind of work is because animals are simply amazing. Thank you.
Ladies and gentlemen, cheer it up! Woo! Give it up for this Space Coast Little Big Band! Woo! On fire! Woo-wee! Oh, I missed the hammerhead. Wow. The next presenter is Dr. Daley Engel. Marine predators such as sharks are vital to preserving the marine food web and the overall health of the world's oceans. Dr. Toby Daley Engel's research at Florida Tech uses a combination of genomics, bioinformatics, field ecology, and modeling to study mating systems, habitat use, and the impacts of climate change on shark populations. Sorry, everyone, I'm getting a little verklempt. She was on Shark Week on Discovery Channel, too. Dr. Daley Engel also works extensively with collaborators from state and federal agencies, non-governmental organizations, and public groups to conduct biomonitoring of Atlantic sharks, understand the impacts of rising temperature on shark movement patterns, and identify novel species using genetic techniques. Woo! But I remember her most as being on Discovery Shark Week. Ladies and gentlemen, give it up for Dr. Daley Engel.
Thank you. See, don't you wish you... Hi. Thanks so much for coming. Thank you, Phi Kappa Phi, for everything that you've done, for inviting me, and for all of your hard work. And I'm here to talk about sharks. So, believe it or not, they're not all great whites, just so you know. We can get that one out of the way right now. Also, the megalodon is definitely extinct. Will not entertain any other questions about the megalodon. So I'm going to talk to you here about some stuff that you may have heard about sharks, and then some other stuff that you hopefully haven't, that's hopefully new to you. So things that you might not know about sharks is that they're actually incredibly ancient as far as animals go. So here you have, this is a timeline of life, if you will, and way back at the bottom, right here, those are where the sharks sort of come off of that tree of life, way down at the bottom, making them one of the most ancestral animals on Earth. Way before there were dinosaurs, there were sharks. And so as a result, they've had a lot of time to get really good at what they do, and they've had a lot of time to kind of adapt and diversify into all of these weird forms that you might not know about because, you know, they're not really the ones that eat people, and so they don't tend to make the news. But believe it or not, these are all sharks, including stingrays, which are really just flat sharks. So something else that you might not know about sharks is that they grow and they reproduce really slowly, just like humans. So this is a baby whale shark. I know! Squeeze, so cute. This is a baby white shark. So they take a really long time to grow up, and it turns out some sharks may actually live hundreds and hundreds of years. Hundreds of years. So they have relatively few babies at a time because, again, they're not like other fish. So your typical bony fish, like your typical goldfish or salmon, will have thousands and thousands and thousands of eggs or thousands of sperm at the same time, all drifting around like tiny, tiny, tiny little plankton. And so if you happen to fish out a bunch of bony fishes one year, there's always going to be those babies floating around in the plankton for the next year. But sharks are live-bearing. They're not egg-laying. They give live birth, just like us. Here's a pregnant human. That's a pregnant sandbar shark. That's a formerly pregnant dogfish shark. So baby sharks actually grow in the mother's uterus, just like humans, and they're born live, either attached by an umbilical cord to the mama, just like us, or attached to a yolk sac. But either way, they develop inside the uteri and they're born live. And there's not that many of them. So like the bony fish will have thousands and thousands and thousands of eggs and sperm at the very same time. And sharks will only have a few. And another thing they do like us, Dr. Nunez gave a pretty amazing presentation on penises that I can't really hope to even approach, except for the fact that sharks have two. Yeah. So sharks have internal fertilization and external genitalia, just like humans, except they have two. So the female has a cloaca, the male has a cloaca, and also these claspers. There's no erectile tissue like in mammals, but there's calcification and internal fertilization. And I heard there were kids coming, so I left off my slide of penetrative shark sacs. Sorry. See me after. In general, though, most sharks are in the mother's uterus attached to a placenta by an umbilical cord, just like us. Or sometimes a yolk. A very few sharks actually lay eggs. That's mostly little tiny, one or two tiny species, and skates. And here you can see there's the little umbilical cords there. So your average reef shark has, I don't know, depending on the size of the female, between 2 and 10 pups a year. And some big sharks can have as many as 150, and other big sharks only have like 2. So it kind of depends on the type of animal they are. But either way, they are long reproducers, including some of the longest gestation times on Earth. I didn't mention that. But sharks will stay pregnant anywhere from about 7 to about 24 months. Yeah, I know, right? 24 months. I have kids. So it's a lot of, because of this reproductive mode, that sharks are in trouble. Because we treat sharks as just another resource that we use in our society and in our economy, just like any other fish stock, because they're very economically important to a lot of people. But unfortunately, they're in trouble. And you might not know exactly how much trouble they're in, but unfortunately, it's quite a lot. And they're not rebounding like other fishes would because of the way they reproduce. So this is a little bit of a busy slide, so I'm going to walk you through it. This is a chart of sharks caught as bycatch in the long-lining fishing industry for tuna. So bycatch is anything that is another species than what you're fishing for. So if you're in a tuna fishery, bycatch is anything that isn't tuna. And so this is showing us declines in shark abundance as measured by bycatch, because they get caught on tuna lines because either they're chasing the tuna or they're chasing whatever the tuna is chasing. So what I want you to notice first is that these are all probably some familiar species. Probably you've seen these animals before. These aren't the weird, creepy, sort of deep water species that I showed you in the beginning. For instance, who knows what this is on the left? Hammerhead, exactly. How about this guy? White shark, yeah, it turns out the great thing, that's a Hollywood thing. Scientific name, white shark. How about C? Can you see the stripes? It's kind of tough. Tiger shark, yeah. How about D? That one's tough. It's just kind of your garden variety gray shark, but it's a sandbar shark, yeah. And the reason it's important is because its meat is very palatable, very tasty. And so in a fishery, in a directed fishery for sharks on the East Coast, this guy made up 60% of all the sharks caught. In fact, if you've been in a grocery store and you've bought shark, it's probably sandbar shark. Even if it's labeled mako shark, usually this is the beast right here. How about E? Who knows what that is? Thresher. Now the band's getting into it. I love it. How about F? A swimming nose. Blue shark. Excellent. We have a ringer. How about G? Mako. H? Oceanic whitetip. Good. You guys know your sharks. So these are familiar species, right? These are charismatic species we've heard of. The other thing I want you to notice is that all of these abundance lines are trending down. When they hit 0, That means that those populations are extirpated, and that's a type of local extinction. So this is in the Northwest Atlantic, and so when those numbers approach 0, it means that there's none of that species left in that particular area. So there might be sharks in the Pacific Ocean of that same species, but they're not in that area. So that's a problem. So why are sharks disappearing? Well, it turns out there's a lot of good reasons. One is their fins are incredibly valuable. Shark finning is actually a growing industry, largely because of economic boom in much of Asia has put something like shark fin soup, which is a very ceremonial, very luxurious dish in a lot of traditional Asian meals, and made into shark fin soup. So, and the palatable flesh, that's a thing. People do fish for sharks and eat them. Sport fishing. Here in Florida, we do a lot of sport fishing. It's in our culture. It's what we do, and it's awesome. However, when done incorrectly, it can have really problematic effects on sharks. In fact, raise your hand if you ever been down at the beach and saw like just a dead shark kind of rolling around in the sand. It happens every once in a while. Yeah, a bunch of hands. That was a shark that was released from a hook and swam away, and it was fine. What we're finding out about sharks is that they stress. You might have heard that stress kills, and that's true. For sharks, though, it's literal, such that just the stress of being taken out of the water, when you take a picture with it before you throw it back, it might swim away fine, but we're finding out that they accumulate so much stress that they pretty much just die. So sport fishing is having an impact that we didn't realize for a lot of years. Bycatch in the tuna fishing industry, you saw the slide previously. That's a big, big source of mortality. But really, this is the primary reason, and it's because of fear. And guess what? There's a lot of really good reasons and a lot of really bad reasons why we're scared of sharks. Can't say I've seen them all. So this is the most natural thing in the world. If I'm ever out and I'm surfing, and I can't see the water underneath me, I'm definitely thinking about, if it's like dark and cloudy and there's nobody else out, I think about that all the time. It's normal because sharks can actually eat you, but they won't. Mostly because we're not their food. So, just to give you a little bit of insight into the reality of shark attack, it's a little different. Only 3 species are responsible for most attacks, and that's white sharks, tiger sharks, and bull sharks. And bull sharks, by far, responsible for more attacks than any other shark. But when I say attack, I'm including things, because the shark attack files include things like, that thing looked like it was dead on the beach, and I touched it, it wasn't dead, and it hurt me. That's an attack. Yeah. I was trying to take the hook out, and I grazed the tooth, and now I'm bleeding. That counts as an attack. So, believe it or not, shark attacks are really rare. In fact, sharks kill between 0 and 5 people per year worldwide. And very, very frequently, those fatalities are outside of the U.S. There's only about 50 attacks per year in the U.S. And we think that there's relatively few attacks, partly because there's good water quality, better water quality oftentimes than there used to be, and sharks can see well, they can smell well, they can hear well, and they can tell the difference between prey and non-prey. And that's because we think that shark attacks are the result of mistaken identity. So sharks didn't evolve to eat people. We're just not their food. Sharks did evolve to eat things like fish, sea turtles, seals, that sort of thing. And their prey has claws and teeth and spines, and it can hurt a shark. And sharks are naturally cautious, long-lived, very suspicious creatures. And so they tend to bite, and then they back off, and they wait for their natural aquatic prey to bleed to death. We humans tend to get out of the water at that point and survive. And so there's oftentimes no shark attacks in the entire US. And I know you've all seen that on the Discovery Channel with the diver in the cage, and the great white is outside the cage. You've seen that, right? Yeah, it's like the money shot, And that cage is thin, and the diver's inside, and the shark is like mouthing the cage, and it really looks like it wants to eat that diver. But sharks can sense electrical fields. They evolved to use their electrosense when they couldn't see and they couldn't smell, like when a fish is hidden under the sand. And they can pick up on those weak electrical signals that that living thing gives off. So imagine what happens to a shark if you drop a giant metal cage into salt water. And, you know, sharks are like dogs or babies. They use their mouths for exploring. So it's like, I don't know what this is. It's trying to figure out if it can eat it. It's probably not even registering the diver that's inside. So things that kill more people than sharks include a lot of familiar objects. I'll give you a minute. Just so you know, I also censored this one for the kids. See me after. Well, so if you're like me, though, and, you know, hearing that something like a vending machine can kill you faster than a shark may not necessarily comfort you in the moment, right? So let me bring you a little more reality. So I just told you that sharks kill 0 to 5 people per year. We kill over 100 million of them. And they live for 100 or more years. So they're not just coming back. 25% of all sharks are at risk of extinction, and those are the ones that we know about. Unfortunately, 45% of all the species that have been identified are what's called data deficient, meaning the studies haven't been done. They may be going extinct, and we just don't know. So that's a problem. If you're worried about sharks, I know I worry. If you see a shark, just don't thrash around. Don't act like bait. Don't swim with your dog. They thrash around a lot. Don't touch the shark. Even if it looks dead, just don't do it. Don't be like those people. Bad idea. And if you like sharks like I do, try to avoid fishing for them. Support laws protecting them. Support shark research, like the research we do here and elsewhere in Florida. Don't patronize businesses that sell shark products because it's pseudoscience. And tell people how cool sharks are. With that, I will leave you with big thanks to my research group and to Phi Kappa Phi for having me, as well as my funding sources. Thanks.
Holy smokes, that was amazing. Nice pick, I like that. So we have one more presenter, but I do want to let you know that at the end of this one, we're going to have them all up here to do a big bow. And if you have any questions that you would like to ask that maybe came during this process, that you should ask then. It's been a lot of fascinating information so far, but I want to let you know that you will have an opportunity to ask a question. So, Dr. Darby Proctor is an assistant professor of psychology at Florida Tech. Her research focuses on decision-making in non-human primates in order to understand the evolutionary roots of human decision-making biases. Specifically, she focuses on spider monkeys and chimpanzees to elucidate the origins of behavioral and cognitive traits in primates, with much of her research taking place at Brevard Zoo. In addition, she is interested in pedagogy and increasing student involvement in research. As a part of this effort, she oversees the psychology undergraduate Roach Lab on Florida Tech's campus, where students can get hands-on experience working with animals. Ladies and gentlemen, give it up for Dr. Darby Proctor.
Thank you. Thanks. So I have a confession, unlike all the rest of the speakers. I am not a biologist. I am a psychologist, and most of the time when I tell people that, they wonder what I am doing in a psychology department because I don't study people at all. But it turns out that psychology actually just means the study of mind and behavior, and other animals have those too. And so I'm what we call a comparative psychologist. I look at the other animal species out there to figure out what their worlds are like, how they see the world, and how they make decisions. But I'm really interested in the non-human primates. So also something people forget is that we are primates too. So we are one of the great ape species, along with chimpanzees, bonobos, gorillas, and orangutans. And as a comparative psychologist, I'm interested in how we became human. And so if we study humans, that's great, and I know there's some other psychologists out here in the audience, but it doesn't tell us the whole picture, because we can only learn what we do now. But if we study our closest living relatives, the rest of the primates, we can learn where the similarities and differences lie in our behavior and cognition, so that we can piece together the evolutionary trajectory of why we behave the way that we do. So to give you a little overview about the primates, they're divided into 4 main groups. So starting with the group that is least closely related to us, are the prosimians. These are things like lemurs. If you guys have seen the Madagascar movie, King Julian. I haven't actually seen it, but my students tell me that's a thing. Then the next furthest related from us are New World monkeys. These are things like capuchin monkeys, spider monkeys. Capuchins are what Ross had on Friends. Or the Night at the Museum movies, and then spider monkeys, which I work with a lot now. So we'll talk quite a bit about the New World monkeys tonight. Then we get to Old World monkeys, which are things like baboons and macaques. You may have seen macaques in the news recently. Apparently there's some running wild around Jacksonville. Yeah, that's a little weird thing. But anyway, and then we get to the apes, right? And again, we're one of those ape species. So tonight we'll mostly be talking about apes and these New World monkeys. Okay, so when I'm looking to figure out where our similarities and differences are in behavior and cognition, so how we make decisions, how we view the world, a really handy way to do that is by using economic tasks. And I know that sounds a little bit weird to talk about economics and non-humans, but basically economic theory provides a nice framework for how we can evaluate how animals make decisions. And that's because if we think about economic principles, we have some nice theories that we can test, right? We should behave relatively rationally. We should look at the costs and benefits of our actions and decide what to do that is best for us, that maximizes our amount of resources. And non-human primates do this in a variety of contexts. So one of those contexts they do that is cooperation. This is a picture of chimpanzees in the wild, and they're on a territorial patrol. So male chimpanzees will get together with the other males in their group, and they will walk around their territory to see if there are any neighboring groups of chimps that they need to sort of fight and chase off. Now, this makes a lot of sense from an economic perspective, because if they were to try to do this alone, they would get beat up by the neighboring groups of chimps. And so it is in their best interests to cooperate, to work together, to engage in these territorial patrols. But we can also test economic principles in the lab. So this is a video of an experiment that was done a number of years ago, and this is a really simple experiment done with capuchin monkeys, so one of these New World monkeys. And the experiment is this. We give the monkey a rock, and the monkey hands the rock back, and we give the monkey a piece of cucumber. And they do this all day for cucumbers. But the key here is we're going to give this monkey on the right a cucumber, just like she's been doing all day, and we're going to give the monkey on the left a grape. And probably to most humans, as well as primates, grapes are a better food item than cucumbers. Okay, so here's what happens. Why in the world would a cucumber that you just ate, you like cucumbers, why has this cucumber suddenly become bad? Why are you suddenly throwing food away? Why are you throwing it back at a human, right? This totally breaks traditional economic theory, which is fine because I don't actually study economics. I study behavioral economics. And behavioral economics is this intersection of economic theory, but paired with how we actually behave. Because just like Lance here, who's rejecting free food, we often do things that don't make sense from an economic perspective, right? I could do the same experiment with you. So I could hand someone in the audience a $100 bill, which clearly I'm not going to. And then I could hand one of you a penny. And whoever gets that penny, you're going to be really upset. And you shouldn't be. It's 1 cent more than you had when you walked in. But we have this sense of fairness, right? We have this feeling that we don't like this. And so this is what I'm really interested in, is these times when we make decisions as primates, both humans and non, where we're doing things that look like they don't make any sense. But if we look deep enough in our evolutionary history, it might be that some of these behaviors make a lot of sense. And that's actually the case with inequity aversion, which is what we just saw Lance objecting to, that unequal payment for the same work. So what we found is that inequity aversion is tied up with cooperation. We think those two things co-evolved. Because if you're going to cooperate with someone, you need some sort of a mechanism to figure out whether or not cooperating with that individual is in your best interests. If you're cooperating with someone and they take all of the reward, don't cooperate with that person anymore, right? It doesn't make sense. And so we think inequity aversion is this mechanism that allowed cooperation to evolve. Now, we see cooperation throughout several primates, including us. We're the most cooperative species on the planet. We'll even cooperate with strangers, which is pretty rare in animal behavior. We also see our closest living relatives, the chimpanzees. They readily cooperate, both in the wild and in the lab. But what's more interesting is this picture when we start looking at these New World monkeys. And so in New World monkeys, we've tested capuchin monkeys a lot. And we've done that with this apparatus that's called a bar pull, which is pictured here. Basically what you do is you give two monkeys that are next to each other access to this heavy apparatus. It takes both of them pulling at the same time to pull it in to get that food reward. And so we can assess cooperation in this way. And capuchin monkeys do this, which the New World monkeys, being more distantly related to humans, are not as cognitively sophisticated as things like chimpanzees and the other apes. And so it's a bit of a puzzle why capuchin monkeys can do this. It might be that capuchin monkeys have this really big brain. So they have a much larger brain to body size ratio than any of the other New World monkeys. And typically the relationship between your brain size and your body size is a pretty decent indicator of how intelligent you are. And so they have this sort of outlier in the terms of the brain to body size ratio with the New World monkeys. But it might also be that we just don't test a lot of New World monkeys. There's a bias in science to test relatively few species because those are the species that happen to be in a lab. So what if we look to another New World monkey? What if we look to the squirrel monkey? So a close relative of capuchin monkeys. And here are the capuchin monkeys, here are the squirrel monkeys doing this type of a bar pull task. So what you'll see here is the researcher. So this research was done by Kate Talbot, who was very frustrated by it because after 8 months, she trained them to do it, and they never understood it. And so what's going on here? Why can't these monkeys, who are really closely related to capuchins, do this thing? Well, it turns out squirrel monkeys don't cooperate in the wild. So maybe these animals have to cooperate in the wild in order for us to get them to be able to cooperate in captivity. So here, we have a unique opportunity to study some of these primate species that aren't well studied. We have access to spider monkeys through the Brevard Zoo. Now, spider monkeys also don't cooperate in the wild. But when we don't see cooperation in the wild, there's always a question. Is it because they can't cooperate, they don't have that cognitive ability? Or is it because they don't need to? There's no problem in their environment that requires them to do that. And so I wanted to test the spider monkeys here at the zoo. So this is essentially the same apparatus that you just saw with those squirrel monkeys. It's just bigger, because spider monkeys are bigger. And this is a video of what happens. So you'll see one of my students walk in front of here, and she's putting pieces of food in those little cups. And we have the spider monkeys have ropes, and they pull in this tray. They coordinate their actions to get at those food rewards. So I had no idea when we started this project if these monkeys would cooperate at all, because they don't in the wild. Well, it took them about 3 minutes to solve this. So clearly, they are able to cooperate in captivity. So the next question I really want to ask is, well, okay, if we see them cooperating, and we think that an equity aversion co-evolved with cooperation to help enforce it, do these guys have an equity aversion? And the current answer is, I don't know, we haven't tried yet. So a bit of a letdown for this story. But, but it leads me into what I want to talk about next. Because one other reason these animals might be able to do this task, even though we don't see them cooperating in the wild, is because of the type of social structure they have. Chimpanzees, humans, and spider monkeys all have the same social structure. We live in fission-fusion societies. That is, we have a big group that we know, and then we break off into little subgroups for days, weeks at a time. And to manage social relationships throughout space and time, you need to have a fairly high degree of cognitive sophistication. And so that might be why we're seeing these monkeys succeed at this task. Now, none of my research would be possible without Brevard Zoo. They have been an essential collaborator in bringing this type of science to Brevard County and letting us do this here at Florida Tech. And recently, the zoo, actually before I say that, I do want to say one word about the type of research that we do. So sometimes when you say you do research with primates, people think you might be doing sort of evil biomedical things. That's not what I do. Everything I do is voluntary with the animals. They can choose whether or not to come up and play our silly games or not. And there's honestly no way I could force a monkey to do anything they don't want to do anyway. And so this is actually a type of cognitive enrichment for these animals. They get to stimulate their brain when I bring them these weird human games to play. And so this is a way for us to keep these animals in captivity enriched and also get really cool scientific information about them. Now, Brevard Zoo is a small zoo. Have any of you guys been to the zoo? Yeah, okay, it's pretty awesome. I'm still puzzled a bit how Melbourne can support a zoo of this quality. And one thing that zoos are doing these days are moving to trying to get more science in front of people. Because it's good for the animals, it helps keep them enriched, and it also educates the public about what it means to be doing research with animals and primates in particular. And so some really big zoos, Lincoln Park, Zoo Atlanta, have done things to get science right in front of the people so that they're educating the public and, again, enriching the lives of these animals. And Brevard Zoo has recently had a huge renovation of one of their areas, the areas with most of their monkeys. They're called Rainforest Revealed. And I'm really excited about this because they've done something unique, something that hasn't been done anywhere else in the world as far as I know, which is we've created not just one area for our spider monkeys and not just 2 areas, but we've actually created 3 areas for our spider monkeys, all connected by, I'm supposed to call them sky bridges, but I always call them tunnels, but they're overhead, so sky bridge makes more sense. But basically what this allows these spider monkeys to do is fission and fuse, just like they would in the wild. So we're going to be able to see this really cool natural behavior here in captivity. And then what I'm even more excited about, because I am an experimental researcher, is that this area over to the right in pink, they've actually built us a testing center so that we can bring the monkeys into individual enclosures so that we can work with them one-on-one, so that I can test inequity aversion with these monkeys, along with other things, but that's one of the first things we'll be doing. And so they're calling this the Animal IQ, the Cognitive Research Center. And so if you come out to the zoo, hopefully within the next 2 weeks, this thing will be up and running. And so in the front part of it, the public will actually be able to come in and sit down maybe 10 feet away from where we're doing the science so that people can really get involved and see what it means to be an animal researcher. And so what we're trying to do is make science part of the zoo experience. And so I'm going to close with this video. This is, on the right, this is a macaque in a lab doing a cognitive task. On the left, this is the exact same cognitive task being done by an orangutan on exhibit at Zoo Atlanta. And so here at Florida Tech and with collaboration with Brevard Zoo, we are on the edge of becoming a leader in zoo primate cognition. So I'd urge you all to come out to the zoo and see these exciting things we have going on. Thanks.
Ladies and gentlemen, thanks for coming. Give it up for the Space Coast Little Big Band. We've got a few presenters in the back. Come on out, professors. You come out here, Dr. Drew. Give it up for Dr. Drew Pollard, Dr. Toby Daly Engel, Dr. Toby Proctor, Kenya Nunes, Dr. Kenya Nunes, and Dr. Michael Grace, everyone. All right. Ready? I wanted to give you guys, that sounded so beautiful. I hate to stop you, but I wanted to make sure everybody had a chance to give these guys their questions. So, can we have some lights up on the crowd here so we can, oh, there you are. I can see you. Does anybody have any questions? Yes. All right, Doc, what for the people who are watching us maybe online or something, Doctor, what is with your hatred for megalodons?
I actually think megalodons are awesome. Ryan speaks, thank you. They are persistently mistaken for being alive. So I do my very best to dissuade people of that. There are also no bull sharks in Lake Ontario. Thank you, Discovery Channel. Let's see, what else? No, yeah, definitely dead. Just very extinct. I love them for a fossil.
That's a good answer. So many fascinating things to think about. What else might you be curious about? Yes. Does Lance ever get a grape?
Yeah, so it turns out it's really important when we're working with highly intelligent animals to make sure that we maintain a good relationship with them. Because if we don't, they won't ever do this for us again. So yes, after these tests, Lance gets several grapes and gets very happy. But because yes, we have to make sure we do that. Otherwise, yeah, they'll never come back and test again.
Yes. Dr. Grace, have you done any type of work with mantis shrimp?
The short answer is no. The reason he's asking about mantis shrimp is they are an amazing animal for so many ways. They have some fascinating behaviors, but they have at least 12 different kinds of color sensors, possibly more. So there are some even more extreme examples. Our work is all on vertebrate animals, but I think mantis shrimp are really cool.
Yes. So, Grace, there are colorblind people and there are glasses that allow people to see color. How does that work?
I don't know. No, I really don't know. I really don't. I should look into that. But it is, it can't be. It's simply not true color vision. The light that comes into your eyes is the spectrum that it is, and you have the sensors that you have. And for a colorblind person, most still have two sensors. They're still only going to pick up a certain array of colors. I think what they do is sort of shift the spectrum a little so you can begin to discriminate certain colors that are hard to discriminate, like red and green. But still, it doesn't fix the problem. And by the way, there are people, it's very rare, who have only one, and some who have none. Extremely rare. And if I may, I'll tell you one other really cool thing just about science. The person who discovered the molecular basis of color blindness, actually color vision and color blindness, was a graduate student when he did it, and he actually studied himself. He took his own tissue, he sequenced his own genome, and he figured out how we see color and the mechanism for color blindness all on himself. Really amazing.
Yes. So I'm going to try to remember that whole question. So there is a, did you hear what she said? All right.
Yeah. No, I did understand you. The question is, in animals that are color changers that may be relatively colorblind, whatever that means in that particular species, how do they accomplish this color change? The short answer is no one knows. They really don't. My guess is something like this is going on. In fact, these, what we call extraocular photoreceptors, they're actually fairly common in the animal world. There was even a study some years ago where people said, we have them. They said we have light detectors in the back of our knees. Now, why in the world you would have those? I have no idea. That has not panned out. But anyway, so we really don't know how octopi and other species like that do this. The hogfish is the very first one in which we figured this out. So it's a great question. Love to know more.
Yes. So Dr. Darby Proctor, the monkeys that you get to do research on, are they the leader monkeys, or are they some kind of slacker monkeys, or are they some in-between monkey? What are you doing?
Yeah, so most of the monkeys really love the type of testing we're doing, and my biggest issue at the moment is trying to get them not to all try and test at the same time. But this new facility at the zoo will really help with that. But what you're really alluding to are the dominance hierarchies that primates and most other social animals have. And we certainly have to take that in consideration when we're testing. So Shelly, the alpha female, always gets tested first, because if Shelly doesn't get the food first, she's just going to steal the food from everyone else. And so what we basically have to do is sort of test them in order of dominance / who wants to play that day. And so the lower ranking animals get tested last. But we'll still do that. And that also means that the higher ranking monkeys, they finish all their tests first. And so we have to do what I call fake testing with them, where they're still going to steal your food if you don't test with them. So we just play some games with them to let them get treats, and then they'll go away and let us test the lower ranking individuals. So right now we can test all the spider monkeys except the 2-week-old baby, who has her 2nd week birthday tomorrow. And our juvenile female, so she's just 1.5, so she's still actually just a bit too small to interact with some things. But aside from that, we can test everyone. Yes, so the question was, is there a bias then that because the younger monkeys are getting tested later, that they could potentially be learning from the older monkeys? So certainly that is a thing that could happen and would happen in certain primates. One of the reasons that it's really neat to work at Brevard Zoo is because we have these animals, especially these primates, that they don't have a lot of research on. So with spider monkeys, in fact, there aren't any studies published about whether or not they can socially learn, so whether they can learn from each other. They totally can learn from each other, but it's not been published yet because I haven't run it, but absolutely they can do that. And so yeah, it depends on the type of test that we're doing, whether or not that matters. So if it's just a test where you're making a decision, well, if they're watching other individuals make decisions, it's probably not going to affect them too much, right? If it's a task where we have to teach them how to do something and that's part of the task, then that matters. And so then what happens is one of my amazing students, there's at least one here, wave Emily, yes, will throw some food and the other monkeys will distract them.
Yes, here in front. So the question, you want me to repeat the question, or do you want to do it? Great.
The question was about shark finning and how it's getting to be, in some places, more of a problem and what can be done about that. The reason it's actually going up in some places is because of that cultural significance and the fact that you have a rising economic class in much of Asia that, like me, when I get a little bit of extra money, I aspire to those things that are the hallmark of the upper class. For me, it would be like really nice shoes. But for a lot of people in certain parts of the world, it's something to have at their daughter's wedding. And so with a more prominent middle class, this sort of custom has become more in reach for a lot of people. And so because it's a customary thing and it's very meaningful culturally to a lot of people, the only thing that's really helping, honestly, is cultural change. So, for instance, the basketball player Yao Ming came out against shark finning, and that was a really big deal because he is such a huge celebrity in China. And so, as more awareness gets sort of paid to shark conservation issues, the more we learn, the more 400-year-old Greenland sharks make the news. And the idea that shark populations are both important and in danger kind of gets around. There's no such thing as shark-safe tuna, for those of you of my generation who remember dolphin-safe tuna. Nobody cares about sharks. So if we can change that sort of cultural fear, then they may receive sort of better protection. But just as an economic force, it's pretty unstoppable.
Yes. Wait, monkeys and what? Oh, monkeys and eyes.
They're essentially identical, as much is identical between us and them. In terms of color vision, humans and some of the great apes have this. The old world monkeys have 3 different types of color sensors, so they can see color very well. Other monkeys, like New World monkeys, they are part of that group that has only 2 color sensors. But you know, if you know anyone colorblind, you can still discriminate colors. So all monkeys have color vision. It's just some have 2 color sensors, some have 3.
Yeah, and the picture with New World monkeys is actually a little bit more complicated than that. So a lot of them have these 2 color sensors. But in New World monkeys, it's very strange because it tends to be different depending on which sex you are. And so in spider monkeys, for example, females have full trichromatic vision, so they have those 3 sensors. And a lot of spider monkey males only have 2. Yeah, I know, it's really weird. And we don't really know why, because for a long time we thought the whole reason primates had these 3 color sensors was so that we could detect the ripeness of fruits and not eat fruits that were either under or overripe, because that could be toxic. And yeah, so the New World monkeys are really blowing that out of the water, and we don't know what to make of it. Yeah, but why would it be sexual? Okay, so Dr. David Ingalls said sexual. Okay, well anyway, yeah, that's true. We're nerds. So yeah, it's a bit of a puzzle, and from a sort of practical standpoint, it's really annoying, because if I want the monkeys to discriminate between something and I want the males to participate, I can't just make things 2 different colors. I have to make it different colors and different patterns, so that we're sure that the males, if they are colorblind, because we don't even know, right? Because it's most but not all, can see that. So it's a really weird thing that I don't get.
We have time for about one more question. Dr. Hammett. Yeah, Dr. Palmer, how do you know that bees see what they see?
I don't know, Dr. Grace. How do we know that bees see that they see? So actually, I will admit, I'm not an expert on insect vision. There's been a number of people who look into this, and clearly insects are able to see into ranges of spectrum that we don't. And then when you look at the pigmentation on the flowers, we see that there's different colors in different places, and many of those colors are at ranges that we don't see. And so I think there's a little bit of artistic rendering, obviously, I think, to some of this, but definitely the patterns that we see are not the patterns necessarily that insects see. So there's a lot of flowers that are single-colored to us, but then when you look at them and you move a little bit beyond the human spectrum, you actually see like landing strips, basically, where it's like, sit down here and collect pollen. And I'll pass that to Dr. Grace if he has any more to add.
Well, I'm not sure how to give a short answer, but I think it's a great question, because what it really speaks to is how we conduct science. Not everything is plainly observable. I talked about behavior versus vision versus brain processing. You can see an animal doing something. You can see that monkey shaking the cage and getting frustrated. But a lot of things you just can't see. So I think in terms of what does a bee really see, it's kind of the same as asking, what's the atmosphere like on that planet? Or any number of things. Looking at molecules in a test tube, you can't see them. So what we do is we rely on lots and lots of research that has come before, so that we understand the basics, and we understand the tools that we have to study things that we can't normally see with our own eyes, and then you just keep pushing the limits a little further, a little further. So there are good ways of knowing what a bee sees. For example, you can sequence the genes that encode the molecules that detect light. And from that sequence, we can determine what part of the spectrum they can actually detect. And then you can also go in and do things like put electrodes, tiny little electrodes, inside the nervous system of a bee and record which colors actually excite the nervous system. So it's a big, broad question, but it really applies to much of what we do in science.
All right, everybody, give it one more burning question. I'm sorry, she's been super patient. Go ahead, one last one.
Well, actually, I didn't mention here, but we are testing in women as well. Because if we can, the whole thing about sexual arousal and erection, well, actually, in women, we don't have something that corresponds to erection, but we know that has a lot of physiological answers that can be stimulated. So it's via nitric oxide. That's the main thing that can cause an erection. So we have a test, our peptide also, our BZ 371, the same topical formulation in men and women as well, and we see results. And it's the same way, the same reason why Viagra is increasing for women. I think for Viagra, doesn't work so well for women as men because it's an oral administration. Maybe a topical administration would be better, but I can see the same pathway working in both. Okay.
All right, everybody, give it a little round of applause. Thanks for coming. Take a bow, everyone. So weird. Don't do that in a classroom, right? All right, thank you for coming, everybody. Hope you enjoyed the night. Thanks for coming, and if you get a recruitment card for Phi Kappa Phi, make sure that you sign up.